Crawford.
Not gonna happen. Not gonna
Clip
happen.
your toenails.
I mean, I'm fine with the setting.
But I don't know what she said.
Yeah,
At what stage is the mitotic spindle formation complete?
complete, like totally set up, 100%. I believe it's either prometaphase or metaphase. I think it's metaphase. Metaphase is when it's totally set up. When is the mitotic spindle complete? Because in prometaphase, like I said, your chromosomes are there, but they're not arranged in any particular order. And so the spindle is starting to kind of settle itself, but it's not completely settled until you have it in metaphase.
Okay, good. When it lines up, okay.
Yeah, when it lines up and when you have all of the microtubules attached to all of the DNA, to both sides of the DNA.
That's metaphase, right?
Yeah, metaphase.
Describe in detail how mitotic spindle functions.
How it functions? Well,
That is it right there. I'm just asking.
yeah,
It's
that's
pretty
it.
simple.
How do the mitotic spindles pull the chromosomes apart?
With the microtubule?
The microtubules shrink back into their centromeres, and that's what pulls the DNA apart. That's how the mitotic spindle functions.
Okay.
You got these microtubules coming out, attached into the DNA, and then when the microtubules shrink, it pulls the DNA apart. It's that simple.
Did you make up the test already?
I am building it right now. It's not complete.
So the answer is not.
Can you build it from some of the questions on the textbook website?
On the what website?
The textbook website.
Oh, I mean, yes, I can. Will I?
Will you?
Just build it from the review.
There are some questions on the review on the test. Like that one with half a cell or half as much DNA, that one's on there.
On here.
From the questions in the book.
The topics that are on that review are the only topics that are going to be discussed on the exam. But just because I don't list every single little vocabulary word that was in all the chapters, doesn't mean you're not going to have to know vocabulary, right? So you're going to have to know what a sister chromatid is.
Oh, that's on here.
And you're going to have to know what a homologous chromosome
Those are
is.
on here.
Can you go over that? I do not know what.
Yeah, I'll go over that real quick and then, or Cameron, is yours related?
What does DNA PLLI do?
DNA polymerase
Oh, DNA polymerase.
is one. DNA polymerase 1, it's just abbreviation. Yeah. All right, so difference between sister chromatids and homologous chromosomes? All right.
That's on here somewhere.
Yeah, it's on the cell cycle on the first page.
DNA replication.
So, we have a cell. The cell has, in G1, before it goes into S phase, let's just say it's got 3 chromosomes. So in S phase, what's going to happen? You're going to replicate your DNA, right? So now you're going to have copies of each chromosome. Well, these copies, those are the sister chromatids. They're exactly genetically identical to this initial piece of DNA. It's just the result of what you get once you replicate your DNA in S phase.
So 2 individual chromosomes are, chromosomes, but 2 matching chromosomes are a chromatid?
So technically, when you're in G1, because remember, when your DNA is uncondensed, it's called chromatin, and it doesn't condense until prophase, which is in M phase. So before it gets to M phase, it's chromatin. Then once you get into mitosis, it becomes chromosomes. So chromatin is the uncondensed version of DNA, and then chromosomes are the condensed version that look like, the X's. And so the sister chromatids, all it is you took a piece of DNA, you made a copy of it in S phase, and they happen to be attached together at the middle, but they're still genetically, think of it as like conjoined twins. Okay, they're the exact same, identically, they just happen to be joined together. Probably a bad example.
Makes sense.
Okay. So the homologous chromosomes, on the other hand, so in mitosis, we're separating the sister chromatids. So we're separating the copies so that one copy goes into one cell and the other copy goes into the other cell. Now, in meiosis, has 2 stages of division. So same thing in G1, we're still going to have this 3 pieces of DNA. In S, we're still going to replicate the DNA. And then in meiosis 1, there's still homologous chromosomes in mitosis. It's just that we only care about separating the sister chromatids. Because in mitosis, you start diploid and you end diploid. So there's still chromosome number 1 from mom and dad. There's still chromosome number 2 from mom and dad. It's still diploid. It's just that instead of separating the homologs from one another, meaning mom to one cell and dad to one cell, we're putting all copies of the chromosomes are being separated. So in other words, in mitosis, if you looked at metaphase, you would see 42 chromosomes, not 42, 44 chromosomes, because the X and the Y chromosomes, they're different. You would see 44 of them lined up. So here's chromosome 1 from mom, here's chromosome 1 from dad. 2 from mom, 2 from dad, 3 from dad, whatever. So you're still replicating your DNA, you're just separating the sister chromatids. Whereas in meiosis, if you look, this is chromosome 1 from mom, and that's chromosome 1 from dad. Chromosome 5 from mom, chromosome 5 from dad. So in meiosis 1, we're separating the entire homologous chromosome. And so that's how we get to this haploid. We're cutting the amount of genetic information in half. So the cell that results is going to be haploid because it's only going to get a chromosome from mom or a chromosome from dad. So let's just say it gets chromosome 1 from mom and it gets chromosome 2 from dad. There's still 2 pieces of DNA, but they're 2 different chromosomes. It's not paired with its match from the other parent, basically. So in meiosis 1, you're separating the homologous chromosomes from one another. So that at the end of meiosis 1, you still have chromosomes present and the sister chromatids are still there. It's just you only have 1 full set of genetic information. Only 1 chromosome from one parent, not 2 chromosomes from both parents. And so then in meiosis 2, now we've already separated the homologous chromosomes, so now the only thing left to separate are the sister chromatids. So that's why we say that meiosis 2 looks identical to mitosis, because in mitosis we're separating sister chromatids. Just like in meiosis 2, we're separating sister chromatids. Yeah?
I know you can go over this for a second. I'm just trying to make sure I understand the difference between the homologous chromosomes and the sister chromatids. So the homologous chromosomes are going to be the result once the DNA is duplicated in S phase. That's how you get the homologous chromosomes.
That's how you get the sister chromatids.
Sister chromatids. So homologous chromosomes would be right when that'd be after S phase, when you're going to duplicate.
Homologous chromosomes are always present.
Okay.
They're always present. The only place that they're not present is in the gametes and after meiosis 1. So to summarize, mitosis, so we said we're starting diploid, and so here's my cell, and I'm going to separate the sister chromatids from one another. But I'm going to end up with 2 cells that are still diploid. In meiosis, I'm starting with a diploid cell, just like normal, except in meiosis 1, I'm separating the homologous chromosomes. And then that's going to leave me with 2 haploid cells because I'm cutting the amount of genetic information in half. Meaning these cells, they'll only have 1 pair of each homologous chromosome. Meaning they'll have a chromosome 1, but it's only going to be for mom or dad, not both. Whereas in mitosis, I mean, there's, like I said, homologous chromosomes are still gonna be there. It's just what pieces of DNA you're separating. In mitosis, you're always gonna be separating the sister chromatids, which are the genetic copies that you get from S phase. And then in meiosis 1, then you're separating those homologous chromosomes that are still there. It's just chromosome 1 from mom and chromosome 2 from dad, sorry. Chromosome 1 from mom and chromosome 1 from dad. So you're separating so that you only get one version of each chromosome, which is why it's haploid. So in a 2N cell, you have mom and dad's chromosome number 1. In a haploid, you only have one of them.
Okay.
And so after meiosis 2, again, meiosis 2, we're separating the sister chromatids. So we end up with 4 of these haploid cells. So if you can remember this, then as long as you use the terminology correct, like you don't want to say that mitosis separates the the homologous chromosomes, because it doesn't. It says separates the sister chromatids. So if there's a question on the test that says, at what point do you separate the homologous chromosomes? The only time you're separating homologous chromosomes is in meiosis 1. And so more specifically, what phase do you actually separate DNA? It'd be an anaphase 1 of meiosis 1.
If I get it right, I get it right. If I get it wrong. Next.
Yeah.
Where's the question at?
There's a couple of them. Go ahead.
Transformation.
Transformation is the process that, so you have a normal healthy cell. It's the process by which that gets converted into a cancerous cell. It's just the name of the event. It's the moment when a normal healthy cell gets changed into a cancer cell. That's a transformation. Or we say that the cell has been transformed. Basically, it's been transformed into a cancer cell. Nancy, do you have a question?
For the main characteristics of the lead-in and lagging strand,
Mm-hmm.
are you looking for, like, the lead-in strand would run 3 to 5? Are you looking
Yeah,
for
so there's
that?
2 points for each one of those.
I mean, it
that I
runs
want you
5
to know.
to 3.
Yeah, so the leading strand is going to be synthesized continuously,
Continuous.
right? Meaning one long piece of DNA. And it's going to be synthesized towards, there's a term that we have not talked about. But so remember, we have helicase coming in here, unwinding it. Well, this place where the DNA is unwinding, this is called the replication fork. Kind of looks like a fork in the road. And so the leading strand is going to be synthesized moving towards that replication fork. Or in other words, it's going to be following behind the helicase. So towards the replication fork. Whereas the lagging strand
It's discontinuous.
is discontinuous, or it's made of Okazaki fragments. You could put discontinuous if you want, but to be technically specific, it's made up of Okazaki fragments. And then it's going away from the replication fork. So if my replication fork is right here and it's moving this way with helicase, the leading strand is going to be this guy, and then the lagging strand is going to be synthesized in segments away from the replication fork. So another way, if you don't want to use replication fork, since we haven't really talked about it until now, you could say the leading strand follows behind helicase, and the lagging strand goes the opposite direction as helicase. If you want to think of it that way, that's fine too.
So you don't need like the 5 to 3 and the 3 to 5?
Because let's see, so here's our 5', here's our 3'. So DNA is, and RNA for that matter, when you're building DNA and RNA, you could only add to the 5' end. And so the leading strand is going to be synthesized based on this 5' to 3' template strand. So that way when you make the new DNA, you're adding to the 3' end, which allows it to be continuous, because you're always going to have a 3' end available, as long as helicase is always unwinding the DNA. But the lagging strand, so since this original piece's DNA is 3' to 5', that means your new pieces of DNA are going to be 5' to 3'. But because we can only add to the 3' end, that's why it has to, you know, add it here and then jump back and then add it there and then, you know, kind of go backwards like that.
Can you just reiterate about the Okazaki, like what that means for the lagging?
Yeah, so the Okazaki fragments are these little pieces of DNA that get made from the lagging strand.
Okay, so they're not on the leading strand.
They're not on the leading strand. So the leading strand is going to be, we're talking about the new pieces of DNA that you're making. So the leading strand is the new piece of DNA that's getting made and following behind helicase. The lagging strand is the new one that's being made, but because of the orientation of the DNA, you're making it in these chunks or in these fragments called Okazaki fragments. Yes?
What's the difference between RNA and DNA?
Some differences between RNA and DNA?
Can I tell you?
Yes, you can tell.
DNA is 2-stranded, RNA is 1-stranded.
Let's just make a list real quick. All right. So we got DNA, and we got RNA. DNA is usually
Double-stranded,
double-stranded. RNA is normally
single-stranded.
single-stranded.
At carbon 2, there's an H on DNA, and then there's an OH on RNA. And
Yep.
then the last thing, the T's, the U's, the G's. The A, C, G.
So these are A, T, C, G's, and C's, and this is A, U, C's, and G's.
That's it.
There's actually a couple more listed in the book. I can't remember them off the top of my head. That's why on the test, I'll only ask you for 3, because I can only remember 3 at any given time. So one's double-stranded, one's single-stranded. And remember, in the nucleotide, we have our phosphate, our sugar, and our base. And so whatever's at this carbon right here is also going to determine if it's RNA or DNA. If it's a hydrogen, it's going to be DNA. If it's an OH group, then it's going to be RNA.
And what about the 4 letters? What are they?
Nucleotides,
Okay.
A's, T's, C's, and G's versus A, U's, C's, and G's, because there's no T's in RNA.
When it says describe the structure of the double helix and what makes the backbone, I might, if it's okay, I'm gonna see if I can use the same answer for another one. CHONP is on the minor group, and then the A's, T's, C's, and G's are on the major group.
What do you mean by CHONPS?
CHONP, those are the main things that make up the DNA helix, right? The size.
I mean, the main elements, yeah, but as far as talking about the specific structure of the DNA, more of what I'm looking for,
Okay.
and I drew this out when we first started talking about it. But basically, the shorthand version is the backbone of DNA is made up of these phosphate and sugars repeating.
Oh, okay. Yeah, I remember that.
So it's a sugar-phosphate backbone. And then the base is attached to the sugar, right? So these 3 things all together make up one nucleotide. And so the bases go into the middle, and the backbone is made up of the sugar-phosphate backbone. And also you need to know that it's anti-parallel. So one strand's right side up, and then the other strand, so I'll still have my base and my sugar and my phosphate. It'll just be upside down.
And the base are the nucleotides.
And then these will base pair, these will base pair, and then that will base pair with the next one. So there's a couple things you need to know about the structure of the double helix. It's anti-parallel. The backbone is made up of phosphates and sugars. And then the bases are held together by hydrogen bonds.
What's the last one?
The bases, the A's, the T's, the C's, the G's, they're held together by hydrogen bonding.
Wait, need to know what the A is and what U is.
As far as structure,
No, what their name is, like A is.
yeah, you need to know adenine, thymine, guanine, and cytosine. You should know those, yeah.
You're saying guarine.
Yeah, I just say A's, T's, and C's, and G's because I say them all the time. It just saves time. But yeah, and we even write them shorthanded. So when we write ATGC, we're talking about the entire nucleotide of A, not just the base, right? We're talking about the phosphate, and then that's going to be linked to a T. So you're going to have another, over here. So we write it shorthand. Just because it's easier to write this than it is to draw this, right? So when we say A's, T's, G's, and C's, yes, that's basically the shorthand saying we're talking about this particular nucleotide. But when you actually draw it out and what it looks like, it's going to be this linked to that, or it's going to look like this.
And for their structure, that's the 2 rings and 1 ring?
Yeah, the pyrimidine and the purines.
So yeah.
So the purines are going to have the 2 rings,
Mhm.
and the pyrimidines are going to have the 1 ring.
And who is who?
And the A's and the G's are I believe those are the purines with the double rings. And then the T's and the U's and the, I guess, C's are going to be the pyrimidines with 1 ring.
TUA is one ring?
TUC.
It just says T's and C's.
Oh.
Because you're talking about DNA.
Well, yeah, okay, you're talking about DNA. Yeah, true. Yeah, you're talking about DNA.
Okay, just before I move away from that, so purines, 2 rings, and that is the
Guanine and the adenine.
GA. Okay, and all the rest are going to be the 1 ring, okay.
Mm-hmm.
What's GO?
G0.
G0, I mean, yeah.
That's the non-replicating stage of the cell. So that's what the cell's in 90% of the time when it's not in the cell cycle. So if it's in the cell cycle, that's only 10% of the time. The rest of the time, it's in the non-dividing stage of G0.
Non-replicated cells?
So 90% of the time, yeah, non-replicating. They're not mitotically active
Okay.
because they're not even in the cell cycle.
Non-replicating stage. That would be a good enough answer, non-replicating.
Non-replicating, yeah, they're not dividing, they're not replicating.